Okay, well, welcome to this short talk, which is from Travis Wheeler, who's at the University of Montana and is an associate professor there. Travis has a strong background in computational biology and did a postdoc at Genela Farms with Sean Eddy. And today he's going to tell us about metagenome annotation and visualization through his poster, which will be available to be seen soon. So the virtual floor is yours, Travis.
Great, thanks, Lynn. Folks in my group develop methods and software for sequence annotation and for visualization of those annotations. And this is a short talk, so it'll be relatively quick. I was selected to give the talk based on the contents of my poster, which is pretty jam-packed. When I submitted that poster, my thinking was, we've got a lot of different projects going on. I'll put a bunch of them on the poster, and then I can talk with visitors about whatever they want to dig into. It's too much for one short talk, so for the purposes of this talk, I'm going to focus on just a couple of aspects of the poster and briefly mention other bits to whet your appetite. You can find me at the poster session tomorrow, or just email me if you want to dig in any deeper. The stuff I'm going to talk about is really the work of some of the great people in my group. I'm putting their pictures here to make that explicit and to keep, and I'll keep the appropriate pictures around in later slides. And while I'm here, shameless plug, I'm looking to hire a couple bioinformatics minded folks, either staff scientists or postdocs in the next few months. And by bioinformatics, I mean driven by the biology and genomics and able to write some code to perform analysis. I'm always also on the lookout for machine learning interested postdocs. So if you think the next few minutes are interesting, and if you can see yourself working on tools like these to analyze genomes and metagenomes, please get in touch. Okay, so I said we work on sequence annotation. What that means is stuff like this. You've got a genome. We want to know what's in it. And you've all seen the results of annotation in genome browsers that show the locations, for example, of protein coding genes in the exons and introns, as I'm showing here. This is taken from the UCSC genome browser. Or you might have transposable elements, which are shown either at the wide scale from the UCSC genome browser above, or represented on our DFAM web server for transposable element families below. These and many more annotations are generated and supported by sequence alignments between a sequence that we want to better understand and a bunch of others that we know something about. And I'm showing, in this case, annotation of eukaryotic chromosome, but it's the same idea for annotating bacterial constituents of microbiome samples and others, that sequence alignments kind of lie at the heart of most of these annotations. And alignments like this are much of the focus of our work, specifically how to produce them more quickly so we can handle massive modern data sets, how to make them more powerful and more effective at overcoming confusing factors like sequencing error, which I'm not going to talk much about right now. I'll hint at them at the end. We also think about how to leverage these alignments in new ways, and in particular, for what I'll talk about today, how to adjudicate between competing annotations. So what I mean by that, anybody who's ever engaged in this kind of sequence annotation knows that you've got a genomic sequence, and you'll frequently find more than one good match to that sequence among the set of sequences that you're comparing it to. And you need to establish some kind of rules for picking which annotations are the winner. That's represented by this picture where down here on the bottom, this shows a fairly simple final annotation. That's the result of adjudicating between a bunch of competing annotations on the top, where a whole slew of different family sequences, for example, may have all claimed, that's a good match to me, and you need to pick some way of picking which one's the winner. And an easy way to choose a winner is to just say, whichever one has the highest score, or whichever one has the lowest e-value, which I think it's clearly the common way of doing it, but I'm going to argue that that's not really good enough for a few reasons that folks who have thought about this will immediately start nodding their heads about. What if the alignments to 3 different candidate annotations all have the same score? Right? Should we just pick one, and what rules should we use to do that? Maybe it's a little more complicated, and those alignments have the same score, but they're different lengths. Now what rules should we use to decide which one is the right one? What if the scores are different but close? Right? Should we be totally committed to one annotation, or should we hedge by establishing some kind of measure of annotation confidence. What happens when competing annotations are overlapping? How do we pick the point at which one annotation stops and the next one starts? What about situations where one element's inserted into the middle of another, as is commonly happens, for example, with transposable elements? How do we pick the boundaries? And just as importantly, how do we recognize that these two components should be stitched together? There are a lot of questions that get answered, and there are a multitude of methods that get used. We've developed a method for computing annotation confidence from a set of pairwise sequence alignments. You've got a stack of sequence alignments, and we can use the scores via a mathy thing that is, this is too hand wavy. There's more details that we can talk about at the poster, but that allow us to be able to compute confidence at the level of windows of sequence, and moreover, we can use those window-based confidences to compute per position confidences based on overlapping windows, and then compute a maximum confidence labeling of an entire sequence so that we end up with a determined labeling of that sequence based on these confidences. And in addition to allowing adjudication directly, we can also just report those confidence values. Along the way, we can identify breakpoints between overlapping alignments. We can recover instances of recombination and gene conversion with quite high accuracy on simulated inputs. And also the method includes a mechanism for stitching together sequence elements that were fragmented by integration of other elements, as I mentioned, is common with transposable elements. So that method is implemented in a tool called PolyA, which is mentioned up here. We're currently working to incorporate PolyA into the Repeat Masker software suite, which is used for transposable element annotation quite widely, and it'll have a wide variety of other applications as well. So I want to take a quick look at an example of PolyA in action. Here, I'm showing a visualization of the annotation of a particular region of the human genome that contains a bunch of nested transposable elements, because that's a natural application of this. This track that I'm showing here isn't from PolyA. It shows the annotation of this region of the genome as presented on the Repeat Masker track of the UCSC genome browser. And these annotations are the result of a tool within Repeat Masker called Process Repeats, which is an adjudicator. It's an expert system that contains a tremendous amount of rule-based logic that's been optimized on human annotation. This next track that I'm showing here shows the results of running PolyA on raw alignments that are used inside of Repeat Masker. And you'll see that it almost perfectly matches the process repeats results without any of the organism-specific or domain-specific knowledge, none of that expert system stuff. It's just a generic adjudicator, which can generalize to other mechanisms, but works well even against a very highly optimized expert system. In the few cases where the tracks differ, we can dig in and look more closely to understand why PolyA made the choices that it did. This is sort of looking under the hood, and we can see, for example, I've got heat maps here that show the confidences and the underlying sequence alignments, which just give a sense of what's going on inside. And I show this largely, it's going to serve sort of double duty of both showing you that there are confidence values on a per position basis that influence the choice of annotation, but also because it shows off a library, a JavaScript library that we've built called Soda. And the genesis of that is that basically for every project we do in the lab, we end up reaching a point where we say, boy, I'd sure like to have a picture of that or to visualize that, either as a part of the actual software release that we're going to give to people who are going to use our software, or just to help us dig deeper into the data. And we've developed this TypeScript or JavaScript library called Soda to make that easier. You can find it here. Soda is not a genome browser. It kind of looks like one up here, but it's more than that or different than that. It's a library that helps us and can help you build interactive web elements that are centered on biological sequence annotations. So you can use it to build browsers, but you could also use it to build things that require more flexibility but kind of break the classic genome browser phenotype. For example, like the way that you can kind of brush over a part of one of these tracks and have a blowout of these heat maps and sequence elements that pop out underneath. All of the elements that live inside of the SOTA library are scrollable and naturally zoomable. They handle standard genomics formats. They can connect to a backend database for real-time updating, all with just a little bit of JavaScript wrapper code around SOTA for each new visualization. I'm going to show one more example of using SOTA just to give a sense of how it differs from what you might think of as a genome browser representation. Motivated by a bacterial genome that includes some phage integrations. So you could draw a linearized representation of a bacterial genome along with some glyphs for bacterial genes and for integration points of phage. You could draw a circular genome, circular representation of that same genome with similar sorts of linked information between the two. And you could even link them so that if you scroll and zoom on the linear form, the circular form will highlight what the corresponding region. And this is mostly just showing off that that feature exists. You could click on one of these glyphs for whichever phage integration you're interested in. They're linked. Either one will do the same thing, and it might pop up a particular chart that shows something complicated. And I'm not going to get into the details of what that is in the interest of time. The key here is that the x-axis is associated with the phage genome, not with the bacterial genome. And you can place other things like protein sequences and PFAM domains that are associated with that x-axis in some way. And these elements themselves can be linked to to impact either the elements here, or you can have a table that itself consists of poly A elements, and clicking on one of these can highlight the appropriate entry in that table. So that's all I'm going to dig in deeply. I'll just mention other stuff quickly. At my poster, we can talk about things like work that we've been doing with a tool called Ultra that is more accurate than alternatives in identifying tandemly repetitive sequence, and also produces meaningful probabilities that we can incorporate into poly A for adjudication, which improves the quality of that adjudication by directly competing tandem repeats versus alignment-based annotations. We've also developed highly sensitive sequence database search software called Fathom that directly accounts for insertions or deletions that induce frame shifts. It produces much better accuracy than other tools, even with really high indel rates. It also substantially improves annotation of pseudogenes and accurately identifies the locations of those indels. And finally, you can, if you come to the poster, you can come and learn about what we're doing to improve the speed of highly sensitive annotation. For example, developing a strategy for sparsely filling in the dynamic programming for performing sequence alignments with profile markup models, which greatly reduces runtime. That's the y-axis here, yet retains a large fraction of the accuracy on the x-axis relative to a tool like Hammer. And with that, I'd like to thank folks in my lab doing almost all this work, the funding sources, and to you for listening. I'm happy to answer any questions you may have.
Well, thank you. I guess I had just a general question about poly A and actually all your tools. Like, what is the fundamental data types that you support? When we think about different kinds of technologies, or is it already assembled and aggregated annotations from NCBI? I just wasn't clear what you're actually aligning and comparing.
It's a great question. So it depends on the tools that we're talking about. So a lot of the tools that we build are in the context of profile hidden markup model sequence database search, which is effectively solving the same problem as BLAST. I have a pile of sequences and one or more other sequences, and I'll compare them. So if they're in FASTA format or a variety of other sort of related formats, those would be accepted for that kind of tool. For poly A, what it's specifically designed to post-process a large stack of results from tools like those. So you get a bunch of pairwise alignments. You've got a genomic sequence running along an entire length of whatever it is, and you'll have little bits that are aligned to candidate annotations that overlap each other, and it can adjudicate between those. You get alignments and what the scoring matrices were that were used to perform those alignments. Does that answer the question?
I was more like Well, yeah, but I was thinking like you don't care then if it's short read, long read, whatever. You're agnostic to the type of data.
Largely agnostic. Yeah, largely agnostic. That the tool I mentioned, Fathom, that performs frame shift aware sequence alignment, is largely targeted towards the scenario where you either got assemblies that were the result of low read coverage, so you don't necessarily have error fixing in place, or you've just actually got reads themselves. They have really high error rates from long reads, and this kind of thing can find metagenomic data sets where rare bacteria will still have interest, but you don't have enough sequence to cure the sequencing error. Does that make sense?
Okay, well, I'm going to try to keep us on time. So thanks again, and I encourage people to go to Travis's poster tomorrow to get more information.